Hi folks, and welcome to this first set of lectures in cell biology. I record these at home in my home office, and my dogs are usually in here with me. They're two Great Danes, Eddie and Henry. Sometimes they'll come over and say hi. Sometimes they'll bark very, very loudly, and I'll have to edit stuff out. I don't try and make these hugely professional. They'll be, I'll misspeak, I'll go back and cover something again, and that's just the easiest way to do this kind of thing. And what will happen is, as you watch through these recordings, you'll see questions that will pop up, and you can answer those questions as many times as you like. You can go back and do them over and do them over, and the scores from those will go to Canvas, and that becomes part of your grade. Now, if you find that the scores aren't going from the lectures, from the recordings into Canvas, and you're not seeing them in there, just wait to the end of the week and let me know, and I can go and take a look on the recording server and get those scores manually. But please don't email me every time there's a little technical thing like that. Let's wait to the end of the week and do it at once. So what we'll do is we're going to go through short sections of material. This first one will be a little bit longer because I'm explaining how this all works. But what will happen is I'll break these up into small pieces. So for example, this little recording is going to have maybe 7 slides, and they'll take anywhere from sometimes 3 minutes to record all the way up to maybe 20 minutes to record. Try and keep them nice and short so they're not too overwhelming. If you have the Canvas app on your phone, you can play these on your phone, you can play them on your tablet, all those kinds of things. So they should be pretty mobile friendly if you want to try and do some studying when you're out and about. I don't cover everything from every single chapter in the textbook, because what I do is I emphasize things which I think are important for a review from general biology, and I then add in things which are new, which are more interesting than some of the other stuff that's covered in the textbook, and I leave out some stuff which I am going to ask you to be responsible for, and I'll put that in a list of learning outcomes each week. It's just there's no sense in me talking over everything. I try and talk over things which are more complex, which are important to be reviewed and covered again. So all that being said, let's get going. We're going to start with chapter 1, which is just some real basic overview and review stuff. There's some slightly new material in here which you'll need to pay attention to. So the first thing I'm going to start off with is cell theory. Now, you've heard of cell theory before. This shouldn't be too kind of revolutionary or too alarming to anybody, because you've seen this before. Cell theory came out of a lot of early work that was based on simply looking at stuff, looking at living things. And that's obviously dependent on microscopes. So you see a microscope on the screen here, we'll talk about that in a moment. So the history of looking at things, just making descriptions of living material, goes back over 300 years, so a long time. And you probably heard about people like Robert Hooke, and he was famous for many things, but one of them was looking at cork cambium under a light microscope, and he described the little chambers that he saw, and he called them cells, and that's where the word cell comes from. It means little room. So when you hear the word cell in a biological context, it means the same thing as if you're looking at a prison cell or a monk cell. It means little room, literally. As what Hooke was looking at were actually dead cells from cork. He did later notice that some cells seemed to contain fluids. He was only able to look at things up to like 30x magnification, so he was limited there. It wasn't until Leeuwenhoek came by sometime later and built his own handheld microscopes, and they gave him up to 300x, and he was the first person to visualize living cells that he called animalcules. And so that was Leeuwenhoek. He was a Dutchman. He was a haberdasher, actually. He was actually interested in looking at pieces of silk and other types of fiber to get an idea of the quality of the materials he was working with. But he discovered that he could look at living cells as well. Advances in early cell biology were really limited by the quality of the microscopes, primarily something called resolution, and that's the ability to see fine detail. That's the ability to tell whether two things are two things or one thing. So the closer two things are together, the harder it is to tell them apart. And so we weren't really, 300 years ago, weren't too able to tell whether we were looking at one thing or two things or 12 things if they were close together, because of the resolving power. So as we got better and better microscopes, here comes Henry, as we get better and better microscopes, resolving power went up, and we're able to look at finer detail. And by the time we got into kind of the 1830s, We had compound microscopes. You have to go away, buddy. Go on. So this on the screen here is a compound microscope. Let's get a pen. I'll sometimes draw things. Compound microscopes have multiple sets of lenses. So those are ocular lenses. You're familiar with the objective lenses, and this microscope actually has a lens under here called the condenser lens. So you know what oculars do, you know what objectives do. Condenser lenses focus the light from the light source, which is down here at the bottom, onto the sample, and then that obviously is then transmitted, refracted and reflected and collected by the objective lens up here. So that's what the condenser lens is. So here's a compound microscope. It must have at least 2 lenses. Here we've got 3. And so as we started building compound microscopes, both resolution improved and so did magnification. And even if you're just using a pretty basic microscope in a general biology 1 or general biology 2 lab, you can get 1000, 2000x magnification with pretty good resolution and see some pretty fine detail. So that really kind of advanced us on to the point where with a good light microscope you can see things which are probably 1 micron away from other things. So we moved a long way in terms of just being able to describe things. The better we got with our microscopes and the more fine detail we could see, we would start to be able to look at structures and see what they were doing, and then we would start to able to look at structure and function relationships. So all in all, really, microscopy led us to what we now call cell theory, which was articulated by Schleiden, Swan and Virchow, and that was based on some work earlier by a guy called Robert Brown, who actually was the first person to identify nuclei in cells in plants, and then Schleiden later then found that all plant tissues actually have cells. Plant tissue is much easier to work with than animal tissue, so a lot of this early work was done on plants and on plant tissues. And then Thomas Schwann of Schleiden, Swan and Virchow made the same conclusion with animal tissue, and that's when we started to get the cell theory coming together. And so building on that later, Virchow kind of completed the picture, and we have these 3 tenets, these 3 main principles of cell biology. All cells come from cells. We'll look at cell division, the processes that produce cells. The cell is the fundamental building block of life. If you're going to look at a tissue, it's made from cells, and then tissues form organs, organ systems, complete organisms, ecosystems, and so on. So that really gave us the bones of cell theory, and of course, even if you're looking at a simple bacterium, you're dealing with at least 1 cell. So that's cell theory, that's nothing revolutionary. Let's kind of just take a step back and talk about resolution before we move on, because this is one of the harder things to comprehend. Getting to the point where we've got to, and the advances that were made since the 1800s, really came down to 2 issues with microscopy: resolution and magnification. Resolution was the main problem, and magnification was secondary to that. Resolution, remember, is this ability to discriminate 2 things as being separate. So if we look at these 2 yellow circles on the left, you can't tell they're separate. They may not actually be circles. They could actually be kind of split here, and there could be a very small gap between them. They look like 2 circles. They are 2 circles, but they could not be 2 circles. They could just be 2 shapes very closely aligned, but we can't tell they're 2 because of the resolution of the image here. Then in the middle here, maybe this is 2 circles, maybe it's not, maybe it's 1 thing. Again, we can't resolve whether there's 1 thing or 2 things. Now, if we get down, if we get over here to the right-hand side, we can see that there are 2 clearly resolved yellow dots, but we can't resolve the fuzzy gray background. So resolution is this minimum distance. As soon as as soon as 2 things become separated by distance, which allows them to tell them as separate, that's resolution. So it's actually a distance. It's usually measured in nanometers or micrometers. And so if 2 things come any closer than the resolving distance, then you cannot tell them as being separate, because we can't distinguish between 1 thing or 2 things. So resolution is actually affected by several variables. The quality of the lens, so how well manufactured are the lenses, what is the material the lens is made from, the media through which the illumination is transmitted. So we can transmit through air, which is what we do in most microscopes that you'll see and that you'll use. You can transmit through water, and you can transmit through oil. You've probably seen it with immersion objective lenses. So oil has got a very different refractive index from water, from air. Air has a refractive index set to 1. Water has that 1.1, 1.2, so not much better. And good oils can get you up to 1.8. And so this is all to do with the density of the media through which you transmit the illumination. In most cases that we'll deal with, that'll be light, but sometimes it'll be electrons being used as illumination. So that's another variable there, is the wavelength of the illumination. The shorter the wavelength of the illumination, the better the resolution. So you're familiar with light having a wavelength function. The shorter the wavelength of the light, if we get down to blue light, that's much better than red light, which is longer wavelength. Obviously, electrons have a particle function and a wave function, and so they have a very, very short wavelength. So that's why you get very good resolutions off electron microscopes. And the last thing is the amount of light that can be collected by the objective lens. So that's a function of how close you get it. You get the lens to the sample. So if you've been in a lab and you've used a microscope and you've got the lens up here and the sample down here, the resolution is never as good as if you change the turret and the objective lens round to the 100x and the sample is now right down close to the objective lens. That objective lens has got better resolving power, probably because you're using oil. The lens is a higher quality, and because it's very close to the sample. That means you can collect more light, and all those things working together give you better resolution. So as microscopy got better and better and better, we started discovering more and more and more cell types. And so here on this slide is just a ton of different cell types. We've got some bacteria, we've got some fungi, we've got some human cells, an egg and sperm, all kinds of different cell types. And as you look at them, I think one of the most important things to take away is that there's always going to be a relationship between structure and function. Let me give you a couple of examples. We'll use 3 examples we'll use off of here. Okay, let's look at the filamentous fungal cells, top left. They're filamentous, they're long, thin rods. The main reason for that is that when you have a long, thin rod and you have things growing in these like stringy finger structures that are called hyphae, you have a very high surface area to volume ratio. Now, that's great if you're a fungus because you want to be spreading your hyphae through the forest floor, through rotting wood, that kind of thing, to collect nutrients. The greater the surface area you have, the more nutrients you can absorb. So that's a structure-function relationship. The Treponema bacterium there, that's corkscrew shaped, and that actually aids its motility, its ability to move in human tissue. Let's have a look. Oh, the sperm in the egg. Sperm's easy. It's got a tail. It's got that flagella hanging off the head. Obviously, that's for motility. So lots of structure and function relationships, and we're always going to be visiting these relationships between structure and function. Now, when you look at these different cell types, let's just compare the sperm here with the retinal neuron down here. Let's assume they're from the same individual. If they're from the same individual, those are some cells out of a total of 40 trillion. And all those 40 trillion cells came from egg fertilizing sperm, forming a zygote. They all had the same genome. So all of your cells have the same genome. Interesting thing about cells, actually, mature red blood cells, this one up here, they don't have a nucleus, so they don't have a genome. But sperm and eggs that you're producing, and your retinal cells in your brain, in your eye, and the heart cells that you have, and the skeletal muscle cells, and the cuboidal kidney cells, and the cells in your liver, they all have the same genome. And so then the question became, well, how can all these cells have these different structures and different functions if they've got the same genome? And that led down a different road that we're not going to get into. This is the realm of genetics, of differential gene expression, of different sets of genes being turned on and off in different cell types to give us different structures. So that's kind of where we are right now. I think as we kind of finish up this little section here, the important messages are that we have a rich scientific history in terms of the development of microscopes and the advances in microscopy. As they became more advanced, our descriptive powers became better, and we became able to start looking at relationships between structure and function. And that's largely what a lot of this course is going to be about, is the relationship between structure and function. So we'll finish this up and we'll move on to section 1.2 in our next video.